Examples of organic acid producers are Aspergillus niger for citric acid, Acetobacter aceti for acetic acid, Rhizopus oryzae for fumaric acid, Clostridium butyricum for butyric acid, and Lactobacillus for lactic acid. Yeast, Saccharomyces cerevisiae, and bacteria are used for commercial production of enzymes. Lipases are used in detergent formulations and are used for removing oily stains from the laundry. Bottle juices are clarified by the use of pectinase, protease, and cellulase. Rennet can also be used for waste to separate milk into solid curds for cheese making. Streptokinase produced by the bacterium Streptococcus and genetically engineered streptococci are used as clot buster for removing clots from blood vessels of patients who have undergone myocardial infarction. Class Cyclosporin A, an immunosuppressant used in organ transplantation, is produced from the fungus Trichoderma polysporum. It is used for its anti-inflammatory, antifungal, and antiparasitic properties. Statins, produced by the yeast Monococcus purpureus, have been used to lower blood cholesterol levels. It acts by competitively inhibiting the enzyme responsible for the synthesis of cholesterol. Recombinant human insulin has been produced predominantly using E. coli and Saccharomyces cerevisiae for therapeutic use in humans. Sewage is the waste generated every day in cities and towns containing human excreta. Microbes are allowed to grow in aerated water. Secondary treatment, they consume major part of organic matter in the effluent and reduce the BOD in the wastewater. A microbial fuel cell is a bioelectromechanical mechanism, bioelectrochemical system that drives an electric current by using bacteria and making bacterial interaction found in nature. Microbial fuel cells work by allowing bacteria to oxidize and reduce organic molecules. Bacterial respiration is basically one big redox reaction in which electrons are being moved around. A MFC consists of an anode and a cathode separated by a proton exchange membrane. Microbes at the anode oxidize the organic fuel, generating protons which pass through the membrane to the cathode, and the electrons pass through the anode to the external circuit to generate current. Biogas is a mixture of different gases produced by breakdown of organic matter in the absence of oxygen. Biogas can be produced from raw materials such as agricultural waste, manure, municipal waste, plant material, sewage, food waste, etc. Biogas is produced under anaerobic conditions when the organic materials are converted through microbiological reactions into glass and organic fertilizers. Biogas primarily consists of methane, 63%, along with CO2 and hydrogen. Methane producing bacteria are called methanogens, and one such common bacterium is Methanobacterium. Methanogens are present in anaerobic sludge and rumen of cattle. In rumen, these bacteria help in the breakdown of cellulose. The excreta of cattle called dung is commonly called gobar. Gobar gas is generated by the anaerobic decomposition of cattle dung. It consists of methane, CO2 with some hydrogen, nitrogen, and other gases in trace amounts. In biogas plants, anaerobic digestion is carried out in the airtight cylindrical tank known as digester. Biomass are collected and slurry of dung is fed into the digester. It has a slide opening which organic metals are incorporated for microbial activity. Anaerobic digestion is accomplished in three stages: solubilization, acidogenesis, and methanogenesis. The outlet is connected to a pipe to supply biogas. The technology of biogas production was developed in India mainly due to the efforts of Indian Agricultural Research Institute (IARI) and Kadi and Village Industries Commission (KVIC). The use of naturally occurring or genetically engineered microorganisms to reduce or degrade pollutants is called bioremediation.